Welcome friends to Grow with Biology. Today's topic is mitochondria. What is mitochondria? A cell cannot survive without mitochondria as it provides energy to the cell. So mitochondria is the energy providing organelle in the cell. When we look at the history, it was first observed by Kohler in 1850 and it was named as Fila by Fleming in 1882. Richard Altman developed a specific stain which he used to stain mitochondria. Then Altman correctly speculated that mitochondria is an autonomous organelle. The present name mitochondria was given by Benda. So the term mitochondria was coined by Benda in 1897, which is an important recognition of mitochondria. Michaelis has proved that and the supervital stains of Janus Green to demonstrate that mitochondria has an oxidation reduction site. Kingsbury suggested that oxidation reactions are taking place in mitochondria, which is a normal cellular process. Otto Warburg, the father of respirometry, showed that enzymes catalyzing oxidative reactions are found in mitochondria. Kenny and Leninger showed that citric acid cycle, which is also known as Krebs cycle, oxidative phosphorylation and fatty acid oxidation took place in mitochondria. In 1951, Leninger also proved that oxidation phosphorylation requires electron transport chain, which is also an important function of mitochondria. In 1961, Michaelis proposed that there is a chemoosmotic coupling theory for the generation of ATP in mitochondria. Now it is when we talk about the location of mitochondria, it is present uniformly throughout the cytoplasm. The orientation is a cylindrical cells. The mitochondria is oriented in the basal or the apical direction, and in leukocyte, mitochondria are remained arranged radially with respect to the centrioles. The number of mitochondria in each cell depends basically on the function of the cell. If the cells require more energy, then the number of mitochondria is high, whereas those cells which require less energy productions, the mitochondria numbers are. Shapes. The mitochondria may be filamentous or granular in shape, depend upon the physiological condition of the cell. Sizes, it ranges from 0.5 micron to 2 micron. When we look at the structures, it is bound by two specialized membrane. The membrane is 6 nanometer in thickness. The outer membrane is smooth and it has some transport proteins which is known as porins, whereas inner membrane is not smooth. It has some enfoldings which are known as cristae. Here we can see that the inner membrane of mitochondria has some foldings which are known as cristae, whereas outer membranes are smooth. The inner membrane has ATPase productions, outer membranes has porins. There is matrix present inside. Now it has two compartments. The outer compartment is known as the perimitochondrial space, which is between the outer membrane of mitochondria and the inner membrane, whereas the inner compartment is found between the inner chamber. The central inner chamber is known as inner compartment or mitochondrial matrix, whereas the space between the membrane on the inner membrane and the outer membrane is known as perimitochondrial space, also known as intermembrane space. The mitochondrial matrix has lipids, protein, DNA. Granules are also prominent. The inner membrane has a cytosols, whereas the cytosols and the M phase, which is known as the matrix phase. Sequestry, which increases the area of the inner membrane, helps in production of more ATP. Shapes. The mitochondria may be filamentous or granular in shape, depend upon the physiological condition of the cell. Sizes, it ranges from 0.5 micron to 2 micron. When we look at the composition, it has protein 60 to 70%, lipids 25 to 30%, RNA 0.5%. Now when we talk about the enzymes in mitochondria, the enzymes in the outer membranes are listed here. They are monoamine oxidase, rotenone, insensitive NADH, cytochrome C reductase, kynurenine hydroxylase and fatty acid coenzyme ligase. In the intermembrane space, that is the space between the outer membrane and the inner membrane of mitochondria, it has adenylate kinase and nucleoside diphosphokinase. The enzymes which are present in the inner membrane has electron transport pathway or electron transport chain, which contain nicotinamide adenine dinucleotide, flavin adenine dinucleotide, diphosphopyrimidine nucleotide, dehydrogenase, four cytochromes, all the four cytochromes which involved in electron transport chain, which are cytochrome B, cytochrome C, cytochrome C1, cytochrome A and A3. All these are present in the inner membrane of the mitochondria. It has beta hydroxybutyrate dehydrogenase and carnitine fatty acid acyl transferase. Now in the matrix of mitochondria, it has malate dehydrogenase, isocitrate dehydrogenase, fumarase, aconitase. All these enzymes are required for citric acid pathway, that is the Krebs cycle. Now when we talk about this function, we find that citric acid cycle will take place inside the matrix of mitochondria, and the inner membrane of mitochondria has electron transport system which will generate the ATP. Now during this biological oxidation of carbohydrate, it releases energy which is utilized by the mitochondria for synthesis ATP. Because mitochondria synthesizes ATP, it is known as powerhouses of cell, because this energy is always generated in the form of ATP. Now this is the electron transport chain. All these enzymes are found in the inner membrane structure of mitochondria. Heat production or thermogenesis. 45% of the energy which is released during oxidation of glucose is captured in the form of ATP, and 55% is either lost or used to generate temperature. Now when we see the biosynthesis of or the anabolic activities, mitochondria perform some of the activities, but mitochondria still contains DNA, and it has lots of machinery which is required for protein synthesis. There is accumulation of calcium and phosphates in the mitochondria of the osteoblast present in the tissues which go calcification. We find that there are large amount of calcium and phosphate accumulated there. vacuum air. So mitochondria is the storehouse for production of this. Thank you.